Fantastic, thank you. I'm really excited tonight to talk to you a little bit about the work that the little company I work for called Caribou Biosciences is doing. So I earned my PhD from UC Berkeley in molecular and cell biology. And while I was still there, my then thesis advisor and I got really excited about a technology that was coming out of her lab. I didn't develop it, but I was around when it was being developed. And she and a few other scientists and I founded Caribou Bio as a place to develop this technology for multiple commercial applications. And so I'll walk you through sort of a high level of what this technology can do and the types of applications that we're developing it for. So the main focus of what this technology can do lives within a field called genome editing. And so what that means is the ability to come into a cell, come into the DNA, and edit it. Change it in very particular ways, so that maybe you're taking out a little genetic material, putting in a little genetic material. It could be a very tiny change, it could be a profound change that somehow alters the ability of the cell to do something. Maybe it changes which proteins it makes, or how many proteins, how much of a particular protein it makes. And so fundamentally what this means is coming in and changing the DNA. And so what is DNA? DNA is the code for all of cellular life. There are 4 building blocks, 4 nucleotides they're called, G, C, A, and T, and together the different combinations of them code for everything, and anything from a simple bacterial cell to us. And so in order to change what a cell does, we have to come in and change the coding, change the DNA within a cell. And so in order to do that, we have to come into the DNA and make a break. That's the first step in being able to edit. You actually have to cut the DNA in order to come in and then do something different at that site. And so what happens when DNA gets cut? Within a normal cell, if you break the DNA, after a little while, the cell will die. So having intact DNA is very, very important for the cell to function appropriately. And in fact, if the cell notices that it has too many breaks, it will actually sacrifice itself for the good of either its larger community, if it's a bacterial cell, or for the good of the larger organism, if it's coming from a human or another animal, something like that. And so for that cell to survive, it has to repair that broken site. And so cells have developed multiple different ways that they can repair breaks. And so I'm just going to summarize 2 of the ways here that are most relevant to genome editing. So one way that can happen is the cell just does its absolute best to glue the broken bits back together again. Now, it's not very good at that, and it tends to make some mistakes as it grabs the 2 broken fragments and glues them back together. And so typically what that means is if you'd started with a perfect sequence, you end up with a couple of changes right at this site. Maybe you're missing a nucleotide, maybe it added a couple extra nucleotides by mistake, but it repaired the break, and so you no longer have a hole in the DNA. And so what that means in the editing perspective is you've just disrupted whatever used to be coded for there. And so if that piece of DNA was coding for part of a gene to make a protein, you've now changed it enough that you're not going to be expressing that gene anymore. And so we call that knocking out. You've knocked out a particular protein so that that cell doesn't make that protein anymore. Now the other option is a little more complicated, but it doesn't cause any errors. And so instead it's a pathway called homologous recombination, which basically means that the cell notices that there's a break here, and it goes searching elsewhere in the cell for pieces of DNA that look like these sequences. And if it finds that, it's able to use it in a sort of copy and paste mechanism. So it's able to say, okay, this looks like this, and that looks like that. So this sequence in here is probably what was supposed to be in here. And it uses this as a template to then fix what goes in here. Now this happens naturally all the time in our cells in particular steps in development. And so typically when that happens, we have, as I'm sure you're all aware, we all have 2 copies of every chromosome. And so it's one chromosome using its sister chromosome to copy from. And so it's actually repairing a gene that's important for us. But in the context of genome editing, we can trick the cells a little bit, and we can deliver pieces of donor DNA that have these important signatures, have the sequences that look like the site that you broke, but it has new material inside it that we can put in here. And so that could mean that we're able to make maybe just a tiny change to how that protein is coded, or this could be a large sequence that actually codes for an entire new protein in and of itself. And we're putting into this cell's genome the ability to express this new protein. And so why is genome editing useful? What does it let us do in the laboratory? What does it let us do in the scientific community that's important? And one of the major things that it lets us do is it lets us study human disease. So the scientific community has done a fantastic job of using all kinds of next generation technologies to look at the sequences, to look at the DNA of healthy people, of sick people, to understand the many different mutations or mistakes that can happen that cause particular diseases, that cause cancer. But we don't have a great way of making those mutations in cells that we can study in the laboratory. And so that's what genome editing provides us, is the ability to take what we've learned about things that seem to be important in cancer or things that seem to be important in particular diseases and intentionally study them in the lab. We can do this in the context of just individual cells that we culture and study at the cellular level. We can do this in the context of animals, for example. So my company, Caribou, has a commercial partnership with a small company in St. Louis called Sage. And Sage uses these types of technologies to specifically make mice that have mutations just like humans who have mutations in particular diseases. And so they're able to create these tools that then basic researchers or pharma companies can use to understand disease, to study disease, and then design, hopefully, new therapies to treat people who have these diseases. So how do we make the cut? How do you edit a cell? How do you make a break at the place that you want and not at other places in the cell? This is very, very challenging, and only in the past 10 or so years has this been possible in the scientific community. And so I'm showing here 3 of these sort of original technologies that people were able to use, called meganucleases, zinc finger nucleases, and talons. The hallmark to all of these technologies is that they require proteins to recognize the very specific sequence in the DNA that you want to cut. And so every time there's a new sequence that you want to cut, you actually have to design an entire new protein, make sure that it can find that sequence in the cell, bind it, and cut it. That's very time-consuming to constantly be designing new proteins to target new sequences, and it's very expensive as well, and I'll get back to that in a moment. And so the cutting-edge new technology that our company is working on, that was invented first at UC Berkeley, and through the help of collaborators in Europe, is based on a little protein called Cas9. And so what Cas9 does is it has the ability to cut DNA, but on its own, it doesn't do anything. It just floats around in the cell and nothing happens. What we need to make it function is Cas9 has a little partner that's a piece of RNA. So RNA is a different type of coding molecule inside of cells. Typically, it's the material that DNA gets turned into that then gets turned into proteins. But there are other pieces of RNA that don't get turned into proteins that are used for other cellular functions, and this is one of those. And so what happens is Cas9 holds on to this little RNA, and the RNA has a sequence that matches exactly the sequence that you want to target for cleavage. And so Cas9, holding on to this little RNA, floats around in the cell looking for its partner sequence. And once that happens, the RNA actually directly interacts with the DNA nucleotides, creating this complex here of the protein sitting down on the DNA. And what happens then is the protein then makes 2 cuts, molecular scissors, if you will, at that particular place in the genome, creating our site-specific double-strand break. And so that means that we can use Cas9, target it to particular places in a cell simply by pairing it with a new little RNA in order to change very specific sites throughout a cell. Now, compared to the other technologies that I just mentioned, where you have to design entire new proteins to target new sites, and this can take weeks to months, for these two technologies and probably years for that technology. To design a new RNA takes about an hour at the computer. You order it and it gets shipped to you the next day. And so this is a very, very fast technology. It's completely changed the way scientists can come up with questions and then quickly test it without having to spend a lot of money in order to make these or buy these. When these technologies first came onto the marketplace, buying one was about $100,000. It was insanely expensive. There were very, very few people who could afford that. Today, if you want to buy one of these, they're about, I would say, $3,000 to $5,000 each. Still reasonably expensive, but there are more labs who can afford that, both at schools like UC Berkeley and at companies. But to order this little piece of RNA, all you have to do is buy a little DNA construct, which costs probably $10, and that lets you make your piece of RNA in order to do this experiment. And so what's been incredibly exciting is we've seen over the past many years people coming up with ideas for what they could do with this technology, or what they could do with genome engineering, but they couldn't afford to do that many experiments. And so now the scientific community is absolutely booming with this technology, and we see lots of people using it in their basic laboratory experiments because it lets them answer questions that they've had for a long time that they didn't have the ability to work on previously. One of the really exciting things about Cas9 is it opens up the opportunity for what we call multiplexing, which is just a sort of obnoxious word to say doing multiple things at the same time. So because all you're doing is delivering the Cas9 protein with a helper RNA, you can actually deliver the protein with several helper RNAs and be able to cut multiple different sites all within a single cell. And so what that means is you can make really complex modifications. So maybe you're studying a disease where it's not just one gene that's mutated, it's 3 genes, or it's 5 genes that are mutated. Previously, if you wanted to make cells that had those types of mutations, you'd have to make them one at a time. It took a long time till you could collect all the mutations within a single cell. And especially if you were trying to make a mouse model of disease, where every time is actually a full generation of mice, or sometimes even multiple generations of mice, you're talking about maybe 5 or 6 years to be able to make one mouse that has those 5 mutations. And so what this technology has now allowed in the research world is the ability to do this in one step. And so I stole this figure from a paper that was recently published by Rudy Yanish, who's a professor on the East Coast, where his lab showed that in one experimental step, they could knock out 5 genes in mouse cells and quickly make a mouse that had all of those knockouts. And so they saved, as I said, many years worth of work and could look at very complex sets of questions that they were unable to ask before. And so, as I sort of alluded to, there's been a tremendous amount of work that's been done. And not only can you do these very simple edits where you come into a particular place and change one gene, or maybe put in a new gene, we can actually rearrange whole chromosomes. So there are some cancers that are caused by a piece of a chromosome, so one of the long pieces of DNA, falling off and reattaching to another chromosome somewhere else. And so that screws up which proteins get expressed when and how, and causes major malfunctions within those cells. And so with this technology, by multiplexing, by delivering multiple of these complexes at the same time, we can actually intentionally swap pieces of chromosomes and make cells that intentionally have those same rearrangements so that we can better understand the underlying biology and why those cells become cancerous. At the same time, it's also let us work with many, many cells in many experiments at once. Since it is so easy to order these little RNAs and to do experiments, it's really increased the capability for people to do experiments. And in fact, a couple of labs recently have started doing experiments where instead of probing one gene at a time, they'll set up, thanks to the help of some robotics and very small volumes of cells, they'll knock out, one at a time, every gene in the human genome, or every gene in the mouse genome, to test at a very broad scale the biological question that they have, or maybe to probe whether a new small molecule drug that they're trying to design has function in a particular cell type. And this technology was first described in the middle of 2012, and here we are less than 2 years later, and I just checked before I came to speak today. There are over 200 scientific papers that have already been published in less than 2 years using this technology. It's been used in everything you can think of, every model organism that's traditional in the laboratory, so single cells, worms, flies, mice, rats, rabbits, and much more unusual organisms as well, including even the silkworm recently. And so it's really opened up a toolbox of genetics, a toolbox of manipulation capabilities that we've had in a couple of cell types before, but we've never had broadly across all of these different organisms. And so it means that a lot of the types of genetics that people have done in other systems before can now be done in lots and lots of different organisms, giving us much more opportunity to study particular questions in the organisms that they come from, which I think is very exciting. And one thing that I'll point out is that using Cas9 to make these specific breaks is really only sort of the tip of the iceberg for what people are starting to do with this technology. So we know enough about Cas9 and how the protein works that we can take the scissors away. We can make sure that it does not actually cut the DNA, but it still uses the RNA to sit down at particular places in the DNA. And so we can use that as basically a homing beacon. We can use that to deliver other capabilities to a particular site in the genome. And so perhaps we want to deliver proteins that will turn on a particular gene so that we express really high levels of it. Or perhaps we want to deliver proteins that will tune down, turn off a protein so it doesn't get expressed as much, or other types of enzymes. And so we can use this as a targeting system to deliver lots of different functionalities. There are many, many labs that are just starting to look at this and all the different ways that they can perturb biological systems to understand how they work. And finally, I'll give you a hint of what I think is to come in 20 years' time, which is directly using this technology to fix mistakes that cause disease. And so what I've done here is I took a figure from a paper that Dan Anderson at MIT recently published, where they demonstrated that they could use Cas9 to correct a mistake in liver cells in mice in a gene called FAH. And so this is a gene when humans have this mutation or when mice have this mutation, it's lethal. It eventually kills them because they can't process a particular molecule. It accumulates and ends up killing either the person or the animal. And so what they've shown is they can actually go into adult animals and deliver Cas9 and a guide RNA that comes in and cuts at that particular site, and in a very small number of the cells is able to repair it and turn it back into the normal sequence and express the normal protein. And these mice then live like happy, healthy mice as if they hadn't had the disease before. Now, I share this with you because I think it's incredibly exciting, but I also strongly caution that it's going to be quite a long time until this technology is actually used in humans. There are, of course, tremendous safety questions that have to be answered. Of course, it's important to make the break at the site that you want, but it's also incredibly important to make sure you're not making breaks at other sites in the genome. And so there is a lot of work that my company and other companies are doing to ensure that the technology is as specific as possible, but also to develop methods to figure out where these breaks are, to make sure that we're appropriately monitoring what's been happening in these cells and that we understand what's been happening in these cells. And so I think with that, I'll just summarize and leave it here. I'm sure there will be some interesting questions. And just remind you that Cas9 is a very exciting protein that with a little RNA partner is able to target double-stranded genomic DNA and make a specific break. And this break can be repaired in a couple different ways, depending on the pathway that the cell uses. In one mechanism, it ends up just gluing the bits back together, causing some small errors, and knocking out, getting rid of the gene that originally was coded for there. Or it can use slightly more sophisticated machinery look for things that have similar sequences and put them back into its genome at that site in order to repair the break. And this is just an example taken from a review where this has been used in everything from bacterial cells up to human cells to engineer them, to understand the biology, and to hopefully come up with better drugs for many diseases. And so with that, I'll acknowledge Jennifer Doudna and Martin Jinek here at UC Berkeley, as well as their collaborators at the University of Vienna and Umeå in Sweden, Emmanuel Charpentier and Christoph Czerlinski, who were some of the first people to describe Cas9, understand how it works, and demonstrate that you could use it in order to do this type of genome engineering. And I'd encourage any of you who are interested in learning more about Caribou and what we're up to, our website is cariboubio.com. So with that, I'd be happy to take any questions.